Welcome to Anatomy and Physiology. This is week 8, and this week we'll be looking at a few remaining topics back from chapter 9, as well as move into material with chapter 10. As we consider the material in this week's short lecture, I've not only taken materials from chapter 9 and 10 of the text, but I've also pulled in material directly from PhysioX exercise 2, focused on some of the major points of physiology I want you to understand in preparation for your lecture exam in this unit. First, we're going to look at the major functional groups of skeletal muscles. That is, we're going to look at the action of a given muscle, determine what the supporting muscles for that action might be, as well as consider what muscles or groups of muscles counter the action taking place. Further, I've pulled from the textbook 4 generalities used to determine muscle action. These are helpful hints or tips to guide you or help you determine whether something is a fixator or an extensor, an abductor or an adductor based on the muscle's relationship to a joint. We then move to muscles as lever systems by considering levers, then looking at specific lever systems, first class systems, second class, and third class lever systems. We consider the myogram and the muscle twitch, that is, a muscle's response to a single action potential from a motor neuron. Thereafter, we're going to look at two major ways by which muscle contraction may be graded, either by considering stimulation frequency or the strength of stimulation. These are two topics you considered last week when you were working on initial PhysioX exercises. We're also going to take a look at the 3 muscle fibers from chapter 9, sharing some general facts about slow oxidative muscles, fast oxidative glycolytic muscles, as well as fast glycolytic muscles, where we find these in the body, as well as how those fibers are used for action. Lastly, we briefly review the 4 factors used to determine the force of muscle contraction, a topic back from chapter 9, as well as material you may want to use this week to complete your PhysioX activities. Let's go ahead and get started. Looking at the action of a muscle or its movement, we find movement is usually the result of several skeletal muscles acting as a group rather than one muscle acting on its own. Most of our skeletal muscles are arranged in opposing pairs of joints, such as flexors or extensors, abductors or adductors. And although a number of muscles may be involved in a given action, the principal muscle involved in muscle action is called an agonist, or it might be called or referred to as a prime mover. Sometimes the agonist is described as a muscle or group of muscles that is most directly related to the initiation and execution of a particular action or movement. We call it the principal muscle involved in muscle action. For every agonist, there's also an antagonist, or a muscle or group of muscles with the opposite action of the prime mover. Antagonists play 2 important roles in muscle function. First, they maintain body or limb position, such as when we hold the arm out or stand erect, and they control rapid movement, such as when you imagine shadow boxing without landing a punch. In addition to antagonists and agonists, we further have another muscle functional group called the synergist, generally recognized as a muscle used to prevent unwanted movement at intermediate joints or to otherwise aid in the movement of the prime mover. Finally, our textbook refers to fixators. A fixator is a muscle or a group of muscles whose contraction stabilizes the origin of the prime mover, such that the prime mover can act more efficiently. Fixators tend to steady the proximal end of a limb while movements occur at the distal end. Now here we have a group of muscles of our lateral arm and a bit of our forearm. When we're talking about action at the moment, we're going to talk about flexing the arm at the elbow joint. And so as we do that, when we consider the primary muscle aiding in this action, we're talking about the agonist, that's the biceps brachii. Now, we also have an antagonist, the opposite, that muscle opposite in action. That would be the triceps. The triceps would help us extend the forearm at the elbow. Now, when we consider the synergist, our synergist is going to help support the biceps brachii by preventing unwanted movement at an intermediate joint. And so we're looking at the brachialis here. We see the brachialis muscle here. This is our biceps brachii. Here's our triceps brachii. Here's our brachialis. Finally, we have the fixator. The fixator is a muscle or group of muscles whereby contraction stabilizes the origin. And so if we look up here at the deltoid, remember we have anterior, middle, and posterior fibers. What we're going to see is when the deltoid contracts, we are stabilizing the origin of our biceps brachii. Moving on, as we consider muscle action, our textbook provides a figure that shows how the action of a muscle can be inferred based on the position of that muscle relative to a joint that it crosses. I call this a cheat sheet of sorts to help us in studying the action of muscles. If you know the origin and you know the insertion of a given muscle in relationship to a given joint, and you know the 4 general rules of muscle action, you can infer what that action, or at least what one of those actions of that muscle might be. And so the textbook outlines these 4 generalities. First of all, muscles crossing the anterior side of a joint produces flexion. So in anatomical position, we look at the muscles that are on the anterior side of a joint to imagine flexion. And some examples of this include the pectoralis major muscle, which if we look at that here, we're going to see that that's located anteriorly to the glenohumeral joint. And so that glenohumeral joint is found right here. And in this manner, we should expect that the pectoralis major muscle provides for muscle flexion, and specifically, one of its actions is the flexion of the humerus. And so we see that here. In contrast, another generality is that of point 2. A muscle crossing the posterior side of a joint produces extension. Now, the textbook in this particular image is going to use the latissimus dorsi as an example, which crosses the back of the glenohumeral joint. And in this manner, we find that this muscle can act to extend the arm. And if the pectoralis major was the agonist, then the latissimus dorsi would act as an antagonist, and vice versa. Next, a muscle crossing the lateral side of a joint produces abduction. Remember the motion of abduction. And if you don't remember these terms, go back to chapter 8 in the discussion of joints. So in our textbook, we see this example here of a muscle crossing the lateral side of a joint to produce abduction. The deltoid is going to cross the lateral side of the glenohumeral joint, where it originates at that lateral margin and the superior surface of our acromion to insert into the deltoid tuberosity on the shaft of the humerus. With this in mind, when we see these muscle fibers contract, specifically the middle deltoid fibers, we see abduction of the humerus. And so we see that here. As these fibers grow shorter, we're going to see that the limb is going to be pulled up or abducted. Finally, a muscle crossing the medial side of a joint is going to produce adduction. And in this image, the textbook uses the example of the teres major muscle. So now we're looking posteriorly at the teres major, and that's going to position itself to the medial side of the joint. So we're going to see this being lateral and this being medial. And it's going to originate on the posterior surface of the inferior angle and the lateral border of our scapula. And in turn, when we see this muscle contract, we're going to see it shorten, pulling our arm back toward the body via that process that we call adduction. And so in this case, contracting this muscle leads to shortening of the fibers of that muscle that results in adduction of the humerus, courtesy of the teres major muscle. Periodically through the course, we're going to bring another science concept in to explain human anatomy and physiology. And in today's lecture, we're going to invite physics in to explain how muscles and bones interact to create movement. And to do so, we rely on levers as an explanation. So first, what is a lever? Generally, levers are a simple machine that helps gain a mechanical advantage, moving an object or applying a force to that object. And as humans, we've been using levers for thousands and thousands of years to help us make our work easier. But not only do we use levers to make life around us easier, we also use levers to manipulate the various joints in our body. And although this isn't a huge topic associated with chapter 10, I would be doing a disservice if I didn't explain how our bones and our muscles work together to serve as levers, thus creating simple machines with muscles and joints. There are 4 general parts that make up a lever, so let's go ahead and review some lever terminology now by looking at these 4 terms. First, again, a lever is a rigid structure First, a lever is a rigid structure that can move around a fixed point. And that fixed point is called a fulcrum, which I'll talk about in a moment. But in our body, our levers are bones, usually limbs, and they're acted upon at two different points by two different forces. That first force, we have the effort, or the applied force, which causes movement. And in the case of our skeletal muscular system, this is represented by our muscle's insertion point along a bone. Then we have the load, or sometimes it's referred to the resistance, which opposes movement. And in this case, our load or resistance is the bone we're trying to move, as well as the tissues, muscle, connective tissue, etc., that goes along with it. Now, let me clarify. Effort is the force exerted by muscle contraction, whereas load is typically the weight of the body part that needs to be moved. And finally, let's go back to this term, fulcrum, right here. The fulcrum is the fixed point, sometimes called a pivot point. And in the case of our skeletal muscular system, our joints are going to act as fulcrums. So with that in mind, motion occurs when the effort applied to a bone at the insertion point is going to exceed the load. Now that we have some general terminology in place, let's go ahead and look at the three classes of levers: first class, second class, and third class. These levers are determined based on the position of the effort, the position of the load or resistance, and the fulcrum in relationship with each other. As we look through these examples, I'll show you what I mean. In a first class lever system, the fulcrum is found between the effort and the load in a traditional seesaw type scenario. And it might be further described as having its axis of rotation positioned between the two opposing forces. And so we can see a general example here. We have our fulcrum, we have load on one side, we have our effort on the other side as we look at the seesaw type structure. Now, when we consider the human body, the function of this kind of lever system, the first class lever system, helps permit a change in direction. And an example of this would be the human skull sitting on the spinal column, which we see here. The upper posterior neck muscles, which we see here, are going to be the effort force, and the resistance is going to be the weight of the skull, which we would see naturally fall down in this position anteriorly. Ultimately, those extensor muscles control the posture of the head within a sagittal plane. So this is a first class lever system where the fulcrum is found between the load and the effort. We also have a second class lever system. And in this system, the load is going to be found between the fulcrum and the effort. And our textbook is going to provide us this very generic example with this wheelbarrow-like situation where we're going to see the load is located between the fulcrum and the effort here. Second class levers are rare in our body. However, we do see an example as given by our textbook. We have the ball of our foot representing the fulcrum, the load, which is going to be the weight of our body, and our effort, which is going to be provided by two muscles, specifically the gastrocnemius and the soleus muscles. Finally, we have a third class lever system. This is the most common lever system in the body, and in this manner, the effort is going to be found between the fulcrum and the load. So last time we saw the load located between the fulcrum and the effort. Now we're going to see the load positioned in one direction, effort closer to the fulcrum. So we see that example here, fulcrum, effort, and farthest away, we see that load. Again, the most common lever system in our body, and our textbook provides this specific example as we're considering flexion of the forearm by picking up a load with our hand. So the fulcrum is going to be our elbow joint. The resistance or the load is what we find in our hand, and the effort is the contraction of not only our biceps brachii, but our brachialis muscles attached to the proximal end of our forearm, providing for forearm flexion and movement of the load. And so this is our third class lever system. Moving along to look at the term muscle twitch and consider this graph we call the myogram is this slide here. This was the topic of focus from PhysioX exercise 2, activity number 1, assigned last week in week 7. And to understand muscle twitch and the myogram, let's go ahead and review some terminology found in our textbook as well as within the introductory materials of exercise 2, activity 1 of PhysioX. First, what is a myogram? Well, if we look here, we're going to see a myogram is simply a graph or a record of muscle contraction, by which we're going to see some distinct phases or periods. We see a latent phase or a latent period, a period of contraction, and a period of relaxation. We also see a refractory period that I'll discuss in a moment. Let's go ahead and define muscle twitch first. So a muscle twitch is simply a brief contraction of all the muscle fibers associated with a motor unit in response to a single action potential from the motor neuron. And there are going to be 4 stages, of which we see here, but I'm going to want you to know a 4th stage. So 4 stages of a muscle twitch as seen by this myogram. And let me reiterate, the textbook only outlines the first 3 phases, but for your exam, if I ask you about the stages of a muscle twitch, I'll expect you to be able to discuss these 3 phases outlined here, in addition to another phase or stage not discussed in the textbook or specifically associated with activity 1 of PhysioX exercise 2. First, we have what's called a latent phase. We see this blue color right here labeled latent phase or latent period. Sometimes it's called a lag phase. This period or this phase is going to represent the time that elapses between the generation of the action potential at a muscle fiber and the start of muscle contraction. During this time, we see calcium ions are going to be released from the terminal cisterns or the sarcoplasmic reticulum. Those calcium ions are going to bind to troponin. That's going to result in the movement of tropomyosin from the active sites of actin, where we're going to see the formation of cross bridges between myosin heads and actin. Tension is going to be exerted and the shortening process of muscle fibers begins. This period is generally known as the excitation contraction coupling phase, and muscle tension isn't yet measurable. This period is very short. We see here very short period of time. In fact, it's usually 1 to 2 milliseconds in duration. Next, we have the contraction period. This is the second phase of muscle contraction, which begins at the end of a latent period and ends when muscle tension is going to peak. It is the period where cross bridges are active. And moving from there, then once we have muscle contraction, we need a relaxation period. This is the third phase of muscle contraction, and we certainly see it as a longer phase. This is the period of time from that peak tension until the end of muscle contraction, and it represents the time where there's active transport of calcium back to the sarcoplasmic reticulum, resulting in a decline in the number of active cross bridges between actin and myosin. Thus, we're going to see a drop in contractile force. Now, I further elaborate by adding a 4th period here, or a 4th phase. This is the period, and we do see it kind of in this region right here. This is the period of lost excitability, where a muscle might receive an action potential, but the muscle has lost its excitability and it simply can't respond to a new action potential for a short period of time. In essence, the refractory period occurs because muscle fibers are depolarized and voltage-gated sodium channels of the sarcolemma must return to their resting state. They must be repolarized in order to respond to a new action potential. We need to move sodium out. We need to bring potassium in order for those voltage-gated sodium channels to once again respond based on muscle depolarization. So again, I do want you to know all 4 periods of your exam, including within the period of relaxation, where we have a refractory period where no further muscle contraction can take place. Next, I want to consider PhysioX exercise 2, activities number 2 and number 3 from last week, where we looked at the effects of stimulus voltage as well as stimulus frequency on skeletal muscle contraction. First of all, muscle contraction can be graded in 2 different ways. We might have an increase in the strength of stimulation. So we increase the voltage of stimulation, causing motor unit recruitment. The stronger the stimulation, that is, the more volts that are applied, the more motor units are activated, and the stronger the contraction. Let's look at the next slide to understand how strength of stimulation aids in motor unit recruitment. First, when we talk about muscle fiber response, muscle fibers follow what we call the all or nothing principle, which ultimately means you have a muscle fiber response or you don't. That's the all or none idea. And what this means is there's no in between. There aren't weak muscle fiber responses or strong muscle fiber responses. If fibers are given instructions to contract, they'll do so with as much force as they can muster every single time. Again, we're talking about muscle fibers, not an entire muscle. Yet, we need to consider what happens at the muscle level, thus going beyond just the muscle fiber level. Recall, muscles are made up of countless thousands and thousands of muscle fibers, as well as are associated with many motor neurons, not all of which receive an action potential at a given time. So with that in mind, we look at these three terms here, the first two of which are going to pertain to muscle fibers, the last pertains to the muscle itself. So first we have what's called subthreshold stimulus. This is the stimulus received by a motor neuron, which produces no observable fiber contraction. We might see graded potential at the motor end plate, but we don't meet threshold to trigger the opening of those voltage-gated sodium channels on the sarcolemma to trigger muscle fiber contraction and depolarization. So that's subthreshold stimulus. Next, we have threshold stimulus. This is the stimulus by which the first observable muscle contraction occurs. And in this manner, we've seen graded potential reach the trigger point of threshold, generating an action potential along the sarcolemma, and nothing can stop it from happening once we meet threshold. Again, these two terms, subthreshold and threshold, don't apply to muscles themselves, just the fibers making up that muscle. And that's because muscles have multiple motor units, such that at any given time, some are going to receive instructions to contract, others remain relaxed. And it's only with maximal stimulus where we see the entire muscle respond. So with that in mind, let's go ahead and define maximal stimulus. Maximal stimulus is the strongest stimulus that increases contractile force of a muscle and represents the point by which all of a muscle's motor units are recruited. So we have the strongest stimulus that increases contractile force by which our muscle cannot respond any further. Next, I want you to recall PhysioX Exercise 2 Activity 2 from last week, where we looked at the effect of stimulus frequency on muscle contraction. Now, in this manner, by increasing the frequency of stimulation of a muscle fiber rather than the strength of the stimulus, you change the rate of firing of the action potential of the motor neuron. And by doing so, you delivered stimuli relatively close together, or we might say a fiber was repetitively stimulated. So let's go ahead and turn to the next slide to look at this in more detail. I want you to go ahead and consider these graphs here. But first of all, recall from an earlier slide when we look at the idea of a muscle twitch. An individual muscle twitch is the mechanical response to a single action potential. And this is a good example of one twitch. So we have one stimulus event, our muscle fiber relaxes, and we have another event. As frequency is changed, these graphs are going to represent the progressive increase in frequency. First, we have what's called temporal or wave summation. This is a phenomenon in which when we have two or more stimuli of the same strength delivered to a muscle in rapid succession, with the second twitch appearing stronger than the first, we see the second twitch appear stronger than the first because the second contraction begins before that muscle has completely relaxed. If additional stimuli continue to be delivered, which we see here, we see 1, 2, 3, 4, we see this stair-step-like effect we call treppe. Next, we have what's called unfused tetanus. So if we look down here at this graph, this unfused or incomplete tetanus, in this manner, if a muscle is stimulated at an increasingly faster rate, so again, frequency continues to increase, the relaxation time between muscle twitches becomes even shorter, and the concentration of calcium ions becomes higher in the sarcoplasm of the cell, and the degree of wave summation becomes greater. There's a phenomenon in which the body maintains this quivering contraction that we see here. So we see that quivering idea right here. Lastly, if stimulation frequency continues to increase, muscle tension will also increase until it reaches maximum. And in this manner, if we look at this graph right here, all evidence of muscle relaxation disappears. So each dip we saw relaxation, even if it was just partial relaxation. We continued to see that here as we hit this unfused or incomplete tetanus. Now here in fused tetanus, we see no evidence of muscle relaxation. So that's going to disappear as the contraction fuses into a smooth, sustained plateau. Now we're going to look at something entirely different as we consider the 3 types of skeletal muscles. We move back to chapter 9 for this. Not all skeletal muscle fibers are equal. They are neither alike in composition nor in function. And maybe you've heard the term red muscle fibers, white muscle fibers. Maybe you've heard slow fibers, fast fibers. Well, muscle fibers are classified into 3 main types, given things like the number of mitochondria, the supply of blood capillaries, the amount of ATP that's being generated. So first, we have slow oxidative fibers. These are the smallest muscle fibers in diameter. They're the least powerful type of muscle fiber. They're dark red because of the large amount of myoglobin and the many blood capillaries found associated with these fibers. Further, they have plentiful mitochondria and generate ATP by aerobic cell respiration, thus the term oxidative. These fibers are deemed slow because myosin heads hydrolyze ATP slowly and contraction occurs at a slower pace. Yet, before you think there's anything wrong with slow oxidative fibers, you should know that they're relatively resistant to fatigue and they can produce prolonged, sustained contractions for many hours. There are endurance fibers, such as what one might rely upon for a marathon, or by which we see our postural muscles of the head, neck, back, legs that require continuous contraction throughout the day. We also have what are called fast oxidative glycolytic fibers, which show some characteristics of slow oxidative fibers, as well as some of this 3rd type that we see, the fast glycolytic fibers. These fibers are intermediate in diameter. They contain large amounts of myoglobin. They're associated with many blood capillaries, and again, they appear more dark red in nature. They generate ATP by aerobic respiration, giving them a moderately high resistance to fatigue. And they're referred to as faster or fast because myosin heads hydrolyze ATP faster than the slow oxidative fibers, usually 3 to 5 times faster, making the rate of contraction faster. And these fibers are very useful for walking or for sprinting. Finally, we have fast glycolytic fibers, the largest diameter fibers with the most myofibrils. And as you might expect from a lot of myofibrils, remember myofibrils are our actin and myosin proteins. They are going to aid in powerful contractions. However, they are low in myoglobin. They have fewer blood capillaries and fewer mitochondria and appear more whitish in nature. They do hydrolyze ATP quickly, thus contraction of these muscles is strong and quick. These muscles are relied on for intense anaerobic movement of very short durations, such as weightlifting or throwing a ball, but they do fatigue quickly. Muscles of the shoulders and arms have a high, fast glycolytic fiber content. They aren't constantly active, but rather when they are active, they're used briefly now and then to produce large amounts of tension. So return back to chapter 9 to consider these 3 types. I recommend putting a little grid together to compare these various different aspects of these 3 muscle fibers. In our last slide of this lecture, I want to review the 4 factors used to determine the force of muscle contraction, taking into account some of the material we've already discussed here, as well as prior lectures, material from the textbook found at the end of chapter 9, as well as materials associated with PhysioX exercise 2. So 4 factors used to determine muscle contraction force. First, we have frequency of stimulation. Now recall earlier from this lecture, the higher the frequency of muscle stimulation, the greater the force a muscle exerts and the stronger the contraction. Further, we have the number of muscle fibers recruited impacting the force of muscle contraction. Point 3, the size of muscle fibers are going to help determine, in some part, the force of muscle contraction. And in fact, the larger the size of a muscle fiber, the greater the force a muscle can exert. Lastly, the degree of muscle stretch plays a role in the force of muscle contraction. When muscle fibers contract, myosin heads attach to actin, forming cross bridges, followed by actin filaments moving closer and closer toward the M line, causing sarcomeres to shorten because we have those Z discs that are going to be brought toward the M line, courtesy of the attached actin filaments. That is what helps produce tension associated with muscle contraction. So, as we look at that, the cross bridge forms only where actin and myosin overlap. Thus, the length of the sarcomere will have an influence on the force generated when the sarcomere shortens. If the sarcomere is stretched past an ideal length, actin and myosin won't fully overlap, and as a result, we'll have less tension being produced. Now, if a muscle is stretched to a point where actin and myosin don't overlap at all, then we don't see any cross bridges form, and no tension is generated. Further, if a muscle fiber is shortened substantially, that overlap between myosin and actin is beyond what we would want to see. Actin filaments can't continue toward the M line. We don't have much ability to move actin toward the midline. Thus, we can't shorten the sarcomeres all that much, and tension is going to drop. And our textbook refers to this as the length-tension relationship, and you will see that in a PhysioX activity this week. So with this slide, this is the last slide of this lecture. It's the last slide of our muscle unit. If you have questions about any of the material I brought up this week, it was kind of a smorgasbord of different materials by which we pulled from Chapter 9, Chapter 10, and from PhysioX activities, kind of a recap of some of the main ideas associated with physiology. Reach out to me during office hours. We can chat about some of these topics. Meanwhile, as always, make it a great day.